Hello, my name is Jacqueline Henshaw, and this presentation is going to be on the influence of phenolic glycolipid 1 in M.leprae on human Schwann cells. And during this presentation, we will cover the basics of what is leprosy and what causes this disease, the basic biology of Mycobacterium leprae, the role of phenolic glycolipid 1 in M.leprae, how PGL-1, which is short for phenolic glycolipid 1, may directly affect Schwann cells, the role of macrophages in M.leprae transport, and how macrophages cause demyelination. And then we'll wrap up with some conclusions. So, what is leprosy? Leprosy is an ancient parasitic bacterial disease infecting primarily the skin and peripheral nerves of human beings. And it's caused by a pathogenic aerobic gram-positive mycobacteria called M.leprae. So some key characteristics of M.leprae. M.leprae is a parasitic straight rod bacterium, as you can see from the image here on the left. It's gram-positive, which you can see from both images here, meaning it's going to stain purple or red. And it has a thick lipid cell envelope. And on this thick lipid cell envelope are phenolic glycolipids. And specific to this bacterium, there will be phenolic glycolipid 1, PGL-1, on the outer surface of the cell. And it will cover some 30% of the cell's surface. So most of the research on PGL-1 focuses on its role in infecting human tissues. Now, most of the study on PGL-1 will go as follows. M.leprae comes into contact and shows a preference for Schwann cells, human Schwann cells, because it can bind with the laminin 2 of the basal lamina. So on the image here on the left, you can see the M.leprae in purple coming in contact with the basal lamina in green. And so this bacterium is attaching to the Schwann cell by attaching to that Schwann cell's laminin 2 receptor. And you can also see an image on the right, image B, depicting the same thing, where the bright red, what you can see right here, these bright red shapes are going to be the M.leprae bacterium coming in contact with the Schwann cell. In this next slide here, you can see on your left a structural depiction, if you're interested, of phenolic glycolipid 1. And on the right, this is another image depicting what your last slide was also depicting, which is, you can see here, this is the cell wall of the M.leprae, and it's coming in contact with the basal lamina of the Schwann cell. And so this was evidence in this research that M.leprae does come in contact directly with the Schwann cell. And then in the next slide, you can see in these electron micrograph images that M.leprae crosses into the basal lamina of the Schwann cell in stages. So here in A, M.leprae is outside of this Schwann cell. This is for the axon right here. And then B, it comes in contact with the basal lamina of the Schwann cell. And then here in C, you can see that the M.leprae has gained entry into the Schwann cell. So the early understanding of how M.leprae infected Schwann cell tissues went something like this. The M.leprae somehow it gains access to peripheral nerve tissue and shows a preference for Schwann cells. It comes in contact using the PGL-1 on the outer surface of its cellular membrane, and it attaches to the basal lamina by laminin 2, the laminin 2 receptor, and then it gains entry into the Schwann cell, somehow proliferates and causes damage by demyelinating the Schwann cell from the axon and then causing axon damage to the neural tissue. This was the widely understood version of how M.leprae infects nervous tissue for a very long time. However, in 2017, Cressida Madigan and her team performed an experiment on zebrafish. And zebrafish had become very popular for studying well, as a host for a lot of studies as it relates to molecular biology, and mostly because they're transparent. And you can see, using fluorescent dyes, a lot of the activity that goes on within their cells. And zebrafish, aside from being transparent, share about 85% of their DNA with humans, so this makes them very good testing subjects. So back to the experimentation of Madigan. Madigan and her team used zebrafish to inject them at the spine, in their spinal column, with M.leprae. And when they injected with M.leprae, the M.leprae was fluorescent. And what they found changed the way that we understand M.leprae to seek out or to move within the body to peripheral nervous tissue. And it also changed the way we understand demyelination and nervous tissue damage to occur. So, when Madigan and her team were injecting zebrafish, they, in this case, had more than one type of zebrafish. There was one that is the wild type that had plenty of macrophages, and then this bottom morphant that was depleted. So it was deficient in macrophages. And what they found is that when they injected the M.leprae bacteria into the injection site of the spinal cord of the zebrafish, these fluorescent bacteria began to travel throughout the body of the zebrafish. And this was, they believed, in large part due to the macrophages that would consume them at the site of injection and then take them throughout the body. They would carry them throughout the body. This is confirmed when you look at the morphant, who does not have very many macrophages, that most of this fluorescent bacteria that was at the injection site stays at the injection site. And the following is a video of exactly that. So the bacteria in blue, the M.leprae, and the macrophages, which are in red, these red blobs here, they're going to phagocytose the bacteria and then begin to move between the myelinated axons, which are green. So we'll watch this video briefly. It's just pointing out one of the macrophages. And you'll see slowly these blue bacteria will begin to disappear. And the macrophages are carting them off. So this demonstrates exactly what they understood to be occurring. Well, let's go to the previous slide here. After the macrophages consumed the M.leprae, they would travel by blood vessels, which you can see here on the right, that this fluorescent M.leprae is being carried away through blood vessels. And so if we look back here on the left, these blood vessels would carry the macrophages, these infected macrophages, out to peripheral tissues where the macrophages would patrol the axons and then eventually slow, presumably because they are infected, and end up stationary on the axon. When they were stationary, they began to demyelinate the axon. And so it blows up right here for you to see that according to this model, the PGL-1 in M.leprae is actually understood to induce iNOS, which is inducible nitric oxide synthase, and this often causes oxidative burst of macrophages. But the point here in this case is that it creates an abundance of nitric oxide, which swells the mitochondria of the axon and causes the dissociation of the myelin sheath. This is depicted well here. This is a rendering. And this shows in box 1, the green here is myelin, and these red dots here are mitochondria. So box 1 corresponds to image 1, box 2 corresponds to image 2. And what you see here, where there has been no demyelination as a result of contact with the macrophage, the mitochondria are normal sized and the myelin has not been, or there's no protrusions of the myelin, And then here in image 2, you will see that there are many protrusions, and the mitochondria here are swollen, which confirms the model depicted from the previous slide. And here, F and G, F is an image and G is a rendering of that exact same thing, but closer. So you can observe in great detail the demyelination and the swollen mitochondria as a result of the nitric oxide. So what does all this mean, and how has it changed the research into M.leprae? Some conclusions that were drawn from her study and also from my research paper is that PGL-1 may bind directly to Schwann cells, but so do other species of bacteria that lack PGL-1. It's not unique for M.leprae to be capable of binding directly to Schwann cells. But her research did answer the question of how M.leprae arrives at these peripheral axons to begin with. And she was able to successfully show that they're carried by macrophages. And this was, in my opinion, and of course, I think in everyone else's, very groundbreaking with regards to understanding how M.leprae affects and arrives at peripheral tissues. And lastly, PGL-1 is believed to influence the expression of inos within macrophages, which leads to the swelling of mitochondria, the demyelination, and the axon damage. And this is very important to understanding the mechanics of how M.leprae actually causes damage and disease in human beings. So what's next? Future research will ideally be directed toward a deeper understanding of the means by which M.leprae proliferates after it has been phagocytosed, and at what stage M.leprae bacteria are taken up by Schwann cells, because they're, just like the previous experimentation has shown, it is obvious that M.leprae is taken up by Schwann cells, but we're not really clear as to when that occurs or how M.leprae proliferates. Does it proliferate after the oxidative burst of the macrophage, or is there another means by which it does that, and is PGL involved in that process as well? That will be great to see in future research. I hope you have enjoyed this presentation and that it's been as interesting for everyone else as it was for me to research. Thank you so much for watching.